I'm going to move on to introduce Associate Professor Glen Westall, who is one of our lung transplant consultants, head of the Paediatric Lung Transplant Service and head of our clinical research program in lung transplant. And Glen's going to talk about defeating transplant rejection and a few other things as well. Thanks, Glen.
Research is going to be absolutely integral to the clinical service that we present. The way that we get better is by working out what we need to do next, and the way we ask that question is by doing research. We're not happy just to follow our management strategies and ask others on how we should get better at what we do, and we're defining how management should be, and we do that through research. The other members of the research team you're going to meet in the next hour, and they're listed there. And I haven't listed them as if there's some hierarchy. The important thing is that they are not at all supported by the Alfred Hospital, and they are here because of the generous support that Lungitude and other supporters have given us. And in many ways, they are the engine room of the research. We do as much as we can in the time we have after we've seen the patients. But these guys are dedicated full-time and really drive the research ideas that we all generate and bring it to fruition. We talk about this hub because it's almost as if we take an executive decision about the research that's being done. There's a lot of research, and in fact research is integral to just about everything beyond the themes I'm going to talk about today. And when we want research, when people are going to do research on our patients, we're going to oversee it. We're going to give you permission to do research because we think it's meaningful research. We think of it's research that's going to improve patient care. And if you're going to come to us and do research on our patients and you guys, we're going to insist that you finish the research. You don't fall through, it doesn't fall through halfway along the way. So we talk about this research hub, but we also made a deliberate decision, firstly, that any research we do has got to translate to improved patient care. And that may not be the same year that we do it, but as Gordon says, the research that was done 10 years ago is what's benefited Wendy 5 years later. And likewise, the research we do today may not benefit the patients who are under our care now, but will do in 5 years from now. The other decision we made early on was that we're not going to try and do everything. We're not going to try and be brilliant at every research project. What we're going to do is we're going to come up with the bright ideas. We're going to come up with the ideas of what we think needs to be done, and then we're going to outsource it. We're going to look around the world and work out who are the experts who can actually answer that question for us. And there are certain efficiencies in that. Invariably, we're using their manpower to do it, and we're using the equipment, for instance, that they may have. So it's both cost efficient and manpower efficient to kind of have this hub and spoke model, and it's been very successful. So these are some of the departments and services that we've reached out, and they're academic, they are other hospitals, and they are industry as well. And we do, the Alfred Hospital is affiliated to Monash University, and obviously that's a large academic centre, and we reach out to the people who are doing relevant research and ask them to do research on our patients. Likewise, and you'll meet Lucy. Lucy's background is in Melbourne University. I did my PhD largely based there as well. And we have lots of very strong links with Melbourne University and the Doherty Institute, which is the Immunology Institute part of Melbourne University. And you'll hear a lot about immunology because in essence immunology is transplantation. The reason transplants work or don't work is what we do with our immune system and whether or not we can control it and whether or not we understand what our immune system is, and that the study of that is immunology. Alberta, we worked out the best people in the field. We're actually a renal team over in the depths of Canada, so we reached out to them and we sent samples out to them, and I'll talk a little bit more about that in a moment. And then we also have industry partners, and I'll say a little bit more about those in a moment. We do recognise that our greatest assets in many ways are our patients, and it's a two-way thing. The reason we're doing this is to improve patient outcomes, but we're incredibly grateful that our patients, when we approach them with research ideas, that they consent to it and agree to give us samples. And the collection of samples has been, again, one of these key strengths. We do multiple what are called bronchoscopies. We look in the lungs 6 times in the first year after anyone has a transplant. And when we do that, we can get a fluid sample from the lung, we can get a tissue sample from the lung, and we can get a blood sample. And all of those are sent off to the hospital for routine analysis, but we also take off a fraction if the patients have agreed and use it and archive it for research. And we can go back to that a year later, 10 years later, and ask questions. And that's a very, very powerful tool for us to be able to research, to have that there. And because we're such a busy unit, 100 patients a year, very quickly we fill our freezers with many, many samples. So we can ask multiple research questions on these samples and do a lot of studies and analysis on them. And that's really allowed us to do our research really quite quickly and efficiently because we have so many patients who are able to study. We're extremely grateful for the support that we have from philanthropic organizations such as Lungitude. Organizations such as Lungitude allow us to do a certain type of research. It allows us to do research that we want to do, but we don't have to ask for the government to ask permission to do it. It allows us to come up with our own ideas. There isn't great funding for small projects. It allows us to do quick projects and create, if you like, building blocks for future research projects. And of course, it allows us to provide salary support so that the ideas that are generated amongst all of us come to fruition because Greg, Bron and I don't have the dedicated time. We can support and recruit dedicated researchers to do it. The other type of support is industry support, and these are some of our industry partners we have. We look for those, but of course there are pros and cons with this. With an industry partner, they of course want some benefit. They want something else from it. So there's a certain we'll scratch your back if you'll scratch ours kind of thing approach to it. They always want something. So there's no point going to a drug company and saying, would you support some research in physiotherapy, for instance? They're not interested in that. They have to see a net benefit. So the cards have to fall out where there's a benefit for both, and that's not always the case. But when there is a benefit, industry support is great, because they like things happening quickly. So they will throw money at something to make something, to a research question to be answered quickly. And that works well for us as well. And these are just some of the examples that we have. So One Lambda is a diagnostic company. One of the forms of rejection we have is when our immune system produces antibodies. And there are not great tools for measuring antibodies. And this is the leading company, and we approached them and said, can we have the new assays, the assays that haven't yet been released, but you want to test it to see if they work? Can we get first access to it? And in doing so, they gave us some assays, and it allowed us to learn more about the immune system before any other centre can. And also, what often happens here is they also will give us salary support so that we can recruit staff to do these studies. This is hot off the press. It's so hot off the press, I'm not sure I can mention it, but we've been talking to CSL, which of course is one of Australia's largest companies, and of course is based in Melbourne. They have a product called intravenous immunoglobulin, which we think would be really helpful if we could offer it to our patients. And some of our patients are on it. It's very expensive. We'd like to offer it to all of our patients. But they want evidence. They want to say, well, it's all very well having an idea of using it. You've got to show that your patients have improved. And we first approached them 10 years ago to do a study. And then for various reasons, including the global financial crisis, they went cold on the idea. But literally in the last few months, and we're in the documents are with the legal departments at the moment, we're hoping to be able to announce many hundreds of thousands of dollars to support a study, a first-in-lung transplant of this drug, a new drug for rejection, which also may also help infection, and hopefully we'll be able to announce that more formally in the next few weeks. And then Greg talked about how we're better at controlling this virus called CMV. And again, we wanted to control this virus better. We recognized there's a company called QIAGEN who had an assay that wasn't released, is not freely available. And we approached them and said, can we use the assay for the first time? And we're hoping to publish data on that very soon. So industry relationships are incredibly important, but they have their pros and cons. There has to be an alignment of interest, if you like. And of course, the reason we could get interest from these guys is that they are never interested if we just present an idea. They want some background data. And what Lungitude and other philanthropic organizations allow us to do is create that pilot data, create a body of evidence that this is a good idea. It's not just a good idea because I'm telling you it's a good idea. We have some data behind this good idea. And these companies want to see data. They don't want to just hear good ideas. They want good ideas supported by good data. And that's when they then sign on to do these big studies, which end up being major investments for them. The other form of support is what's called NHMRC. So this is government support, and it's federal support. And this is, in essence, how all research is supported in this country. But the problem is it's ridiculously competitive. So all of the scientists all around Australia every year go through an annual process of putting in a request for funding. But it's incredibly hard to get funding, and the success rate is of the order of 1 in 8. And Australia is full of great scientists. And every year, sadly, they have to go through this process. But 7 out of 8 of them are being knocked back, saying you can't do that research because we don't have money. But we go through a process of doing that. And the reason I particularly mention it is that, we're delighted to announce publicly for the first time that last year was really a very, very successful year for us, and we were awarded 2 NHMRC grants, which was the first time we'd been awarded one for about 10 years or more. Given we're so happy, we're going to spend a few minutes just talking a little bit about those grants, and already you can see the kind of money that's involved. So what this study allows us to do is going to be done with $700,000 of support, which will be provided over 3 years. So the title, it always has to be nonsensical, but here we go. Extending life after lung transplantation, driving the structural and immunological drivers of chronic lung allograft dysfunction. What that really means, we want to better understand why the immune system causes rejection in some patients. But we wouldn't have got the grant if we wrote that. So this was a grant that I put together with Lucy, who you're going to meet soon, and someone called Dimmy, who works in the Department of Immunology here at the Alfred. I'm going to just give you a little bit of science. Please ask questions or fall asleep if it's too heavy. But this is what we're going to be doing with this grant. The first thing we want to do is we understand we get rejection because our donors and recipients aren't the same. The only scenario that doesn't occur is if you've got an identical twin and they offer you an organ. In every other scenario, there are differences between all of us. What is it, women from Mars, men from Venus? Well, it kind of plays out in transplantation as well. It's our differences that drive this rejection process. Our immune system sees that when you put somebody else's organ in, they see it as foreign and they try and get rid of it. So we wanted to define a better way of determining, what are these structural differences that drive this immune system. And then what we wanted to do is then see how the immune system responds in transplantation. And then we wanted to get some biopsies from our patients and find a new way of diagnosing rejection. I'll say a bit more about that. So the first one is, how do we define differences? What is it that drives rejection in transplantation? It's all to do with this molecule called HLA. We all have these on the surface of every one of our cells, and my HLA is different to Greg's, which is different to Bron's, and it's these differences that our immune system says. It sees an organ and says, hey, that HLA is not my own, that's someone else's, therefore I need to get rid of it, rejection. And we've known about this for 50 years or more. But what we've actually come to recognize, and if you just look at this, we recognize that it's not the whole molecule that's important, it's just these tiny little bits here that's orange, and this is a new concept, but the immune system doesn't see the whole thing, it just sees a very, very small part of it, and that's important. And we've learned a new technology of just recognizing those orange bits rather than the whole molecule. I use this as an analogy, of course, we all know about toll roads, and the toll roads is a bit like the immune system, or the camera is a bit like the immune system, but these cameras aren't seeing the whole car. What they're actually seeing is the number plate, and that's how the immune system works. The immune system isn't interested in the whole structure, it's only interested in that very, very small structure. And we're now using this new way of defining differences between our donor recipients and see if we can better match our patients. And we reported on this as a pilot data and had this published in a prestigious journal just 18 months ago. And this work was supported by Lungitude work from the year before last. And on the back of that, we were able to put this into our current proposal. And what we're aiming to do is recruit a PhD student who will basically take this to the next stage and hopefully make it clinically applicable such that we use this to define who we should transplant any one night. Can we get a recipient who's very, very similar to their donor by
So are you saying that something that Lungitude funded 2 years ago is a direct?
Yeah, so again, what happens with NHMRC, they were, you can write a great idea on a piece of paper and they'll put it in the bin. They want some data behind your idea. And what Lungitude allowed us to do in this paper was create some pilot data that we could show that had been published, had interested the community, and the federal government says, this is believable. And it's that kind of data that then leads to these major funding, if you like. So in essence, the theme in all of this is that the pilot data that Lungitude allows us to do, the concepts it allows us to explore, attracts bigger funding. Success breeds success, if you like. There's two other parts to this. The other bit, as I said, is looking at how the immune response. The immune response is on our cells, and in particular the white cells. There are lots of different white cells, neutrophils, macrophages, T cells, B cells. They all have different roles. But in transplant, we have an interest in something called the B cells, and Dimmy, who's a co-investigator of that, is going to be recruiting someone and has recruited somebody who will be supported for the next 3 years to define how B cells operate in our transplant patients. And Lucy, who you're going to hear about, She has an interest in another immune cell, another white cell called the NK cell, and she has recruited 2 medical students and hopefully a PhD student, and work that she'll be doing to, for the next 3 years, be looking at this, and again, based on pilot data that came out of Lungitude. And then the final part of it is how we define rejection. The way we define rejection, and the reason we do these bronchoscopies, is every time we do a bronchoscopy, we get a tissue sample, we send it down to our pathologist, and we give them, they have a look at something like this. And if there are too many little blue cells in there, they say that's rejection. And that's how they did it 25 years ago, and it really hasn't changed in 25 years. But we recognize that that's not really a very good way of, it's totally oversimplifying what actually is happening. What's actually happening is our whole immune system is fighting this new lung, and there are many, 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 many cells involved in it. It's not just one little blue cell and it's there. There are many, many cells. And what we've come to recognize, there are better ways of getting an impression of that than this. And this involves what's called gene technology, and in particular gene technology. And what we do is we take a little one of these biopsies, we put it in a fancy machine which is incredibly small, and it tells which are the genes of the immune system that are switched on. And it gives us a phenomenal amount of information that we then have to put into a computer, and then the computer in essence gives us spits out an information or a report that says that our patients have no rejection, have rejection caused by a T cell, rejection caused by a B cell, or there's an injury pattern, maybe there's infection there. And it gives us a lot more information. These are the kind of, I've just shown you, this is what our pathologists are looking at the moment. But this is what we're going to be getting back. When we take our biopsies, we're going to get these ridiculously complex readouts that tell us exactly what our body's immune system are doing. And I'm not going to spend time going through it, but it's really putting our diagnostics into 2018 as opposed to 1985 when they first came up with this, and this will be the third key part of this study. And of course, this is what's called the molecular microscopic diagnostic approach, and again, our early work in this, and this is a collaboration with Canada, this is this group in Edmonton, we realized that these were at the cutting edge of doing this. They were doing it in kidneys, and we said, can we jump on the bandwagon and do it in our lungs? And Lungitude funded us to set that up. And send samples over to that, and the government was so interested in that, they said, you've got to take this to the next level, and they've given us funding to do so. So that's the first of the NHMRC grants, and that will be starting hopefully next month. The other one is Ben here. I don't think he is. I was hoping he could come here. Ben Marsden is a guy we've been talking to for about 18 months or so, and he put together a grant with Greg, with myself, and with one of our IB physicians, and again, serious money, $600K over 3 years. You like that title? Clinical implications of the trans-kingdom microbial interactions in the transplanted lung. We want to know about infection in the lung. Again, that wouldn't have been sexy enough. So this sounds kind of vaguely Hunger Games-ish, I think. But Ben's an interesting guy. He's a Kiwi who's literally arrived in Melbourne 2 to 3 weeks ago via spending 15 years in Switzerland. And he's a scientist. He's an immunologist. And his interest is what's called the microbiome, and I'll say a few words about that. But we started talking to him 18 months ago and said, you've got to do your cutting-edge stuff, but in our lungs. And again, the pilot data that was generated interested the government to give serious funding for the project. So the next 3 slides are from Ben. So and it's a pity he's not here because he could present this far better than I. But the concept of the microbiome is that we are, and you may have read about this, is that we're all full of bugs. And these bugs are actually integral to our health. An example of this is when you take antibiotics and you get diarrhea, it's because you get rid of all these bugs that should be there, all these good bugs that are in your gut. And that's why people talk about probiotics. Let's get some good bugs back in there. So there is this sense that we are full of bugs. And that's wrong. It's not a sense, it's true. And in fact, this is Ben's slide. Apparently there are 100 trillion bacteria. So I don't know why we bother just washing our hands with the alcohol wash. We should be showering in it, Ben tells us that there are 10 bugs to every one of our human cells. So in fact, we're only 10% human. We are 90% bug. So, and the reason this is a relatively new discovery, because this is how we used to grow stuff. This is how penicillin was discovered. You take a swab and you put it on an agar plate, and 3 days later you see that something's grown. There's infection there. That's a very primitive way, and most bugs don't grow like that because most bugs don't like growing on this stuff. when you start looking for the DNA, you actually realize that there are, as he said, trillions of bugs and viruses and fungi in our body, and they're absolutely integral. And if we upset the balance, it affects our health. And there's an obesity theory, but some people haven't got enough of the right bugs, and that's one of the reasons why they put weight on. So he talks, and our lungs are full of these bugs, and they need to be there, they're good bugs. And he talks about that here's a healthy, using the tree as an analogy for the lung, there are lots of different classes of viruses and bacteria, and this is a healthy tree, and they're all in balance. But if the lung becomes unhealthy, and you get what's called dysbiosis, that the bugs disappear. And we know that in transplant, we give our patients lots of immunosuppression, we give them lots of antibiotics, and the question we asked is, can we look at all the bugs in our transplanted patients and work out, is it a healthy environment? And if it's not a healthy environment, this is why some of our patients are rejecting it. And again, the government love that idea and have committed to serious funding over the next 3 years, and that's a project that's starting again next month. To wrap up, I'm just going to mention a few other themes for completeness. Our patients love talking about stem cells, no matter whether you're what area of medicine you're in. The first question patients always ask you, have you got stem cells for me? It doesn't matter if you're talking about a broken leg or a heart attack or whatever. What about stem cells, doc? Well, that fixed me. Well, we're using stem cells. Quick, announce it, tell everyone. So this is where we've actually jumped on the bandwagon. This was actually an idea that came from our lung transplant colleagues up in Brisbane, and we work actually very well. There's 4 lung transplant centres in the country. We work out very well. But we are now using stem cells as part of a study. It's not standard treatment, but as part of a study to see if we can stop patients developing worse lung disease if they get chronic rejection. And this has only just started, but you'll be hearing more about this. This is really just a shopping list. I've given you some themes here, but as we've said, we embed research into everything we do, and I think the vast majority of all of our staff, whether it's the nurses, the physiotherapists, the pharmacists, they embed research into the clinical approach they have to our patients. And this is really just a shopping list of a whole series of the other projects that are going on, and I'm not going to go through them, but it's really just to give you an idea. There is a huge amount of going on, and we're extremely grateful for our patients as ever to buying into it. And I think they buy into it because they understand the background and why we're doing it. So I think I finished there. I think Paul Kelly couldn't sum it up better, but from little things, big things grow. And I think, as Gordon says, what starts as a simple idea, before you know it, attracts serious federal funding, and that's why Lungitude is vital to what we do. We're not going to stop asking for support from Lungitude because we're already thinking of the next study for 3 years from now that the government's going to be hooked into. And we can't do those little studies, pilot studies about Lungitude, so I thank you all.